Medurstand Understanding Medicine.
I'm Professor Aziz-ur-Rehman, and today we're going to start a series of lectures, just two lectures, on this very important topic, which is new science. And the topic is a little funny. What controls you? Is it your genes, which we always believe that every function of our body is controlled by our gene, or is it your shit? Please excuse me for this little, uh, maybe not exactly right word, but I actually refer to microbiota, our gut bacteria. I thought it will be a little catchy topic, and I think you will be interested to know what is inside. Uh, the first lecture is basically introduction, the role of human microbiota in health. But of course, the main lecture would be the second one, which is about the possible role of microbiota in different diseases. So it is very strongly recommended that after you watch this video, you must watch the other video also to complete the module. Now, this is part one, the role of microbiota in our normal health. You know, it is generally believed that infant in the womb is sterile, although the latest data is indicating that perhaps that environment is also not completely bacteria free. Some remnant of bacteria, DNA, and other remnant have been found in the amniotic fluid also, but generally we believe that we acquire the microbiota at the time of birth. Now, this is in the womb, no bacteria or very few, no live bacteria at least, and this is in the two placenta, some bacterial antigens come to the infant, and then at the time of birth, also depending upon the mode of birth, is it vaginal delivery or cesarean section, the new bacteria are acquired. Now, this will be actually dependent on the mode of delivery. If this is vaginal delivery, then the commensals would be the maternal vaginal commensals, and if this is a cesarean section, then it could be the skin commensals of surgeons or nurses or the hospital. And then these bacteria grow in infancy, and as the infant is exposed to the environment and different environmental pathogens, antigens, allergens, the immunity develops and the bacteria, they colonize. And at the age of 3 years, they become fully populated, and then they become sort of stable. Although at later also there may be a little bit of perturbation due to various diseases, but generally after slight disturbance they come back. So this is prenatal, neonatal, and postnatal factors. You can see the feeding, whether the baby was breastfed or formula fed, or geographical location, of course, family members, the commensals of family members, then host interaction, maternal diet, and weaning. All these would determine the microflora of the child. And one thing which plays extremely important role is the exposure to dirt. So although this slogan has been taken from an ad, I'm not trying to advertise that particular product, but the fact is that exposure to dirt is important in the development of our immunity. And there has been studies that those children which are brought up in a super hygienic state, they may be more vulnerable to certain diseases like autoimmune disorders and allergic disorders. So of course, too much dirt would be of course bad, but little bit of exposure to these things could actually help in development of our immunity and normal microflora. Uh, Almost all body surfaces and all body hollow organs and entry points, they are colonized by bacteria. But the most abundant bacteria and other microorganisms, also some fungi, also some viruses, some primitive bacteria, they are present in the gut. some are present in the skin, in the vagina, and all body orifices, even the nasal cavity, the ear vestibules, and this every orifice of body is actually has its own bacteria. Even conjunctiva, eye is also not sterile. So, and there are different, these pie diagrams, they indicate distribution of various types of bacteria. Of course, I'm not going to go into the detail. I actually do not know what all this means, but all this, what is relevant is that all parts of the body, they have different type of microflora, and our most of the discussion would be focused on gut microflora. Now, as I said, in the infant, microflora develops, and then at the age of when the child is of 3 years age, it becomes stable. And then it remains stable throughout. Everybody has a signature microbiome, but that remains stable for almost whole life. There may be some perturbation because of some infection, but there is only transient change in the pattern, and then they mostly come back to normal, with some exceptions. You know, like in Clostridium difficile infection, there is overgrowth of some pathogens, and normal flora is not restored. But in most situations, our normal flora is restored after whatever normal flora that for that particular person is restored after slight perturbation due to some infection or some environmental factor. Uh, Human microbiota is considered to be an essential organ because our body cannot function without microbiota. In fact, if I say life is not possible without microbiota, it won't be an exaggeration. And uh, collectively, they constitute like like as big as a brain. I'll just show you the picture. Now, just few numbers. This would be very interesting. 13 trillion human cells. As an average normal person has 13 trillion human cells and 130 trillion bacterial cells. That means 10 times. So if we count the number, and in this era of democracy, we must respect numbers. So in our body, we have 13 trillion human cells and 130 trillion bacterial cells, 10 times more. And almost 1,000 bacterial species and many other species. And these species are cannot be characterized by our traditional methodology. There are some new tests actually which can identify these bacteria as distinct species, and that is how the number is so high. Approximately 1,000 bacterial species are present in human. 20,000 human genes. We have just little over 20,000 genes, which have, of course, different functions. And against that, we have 2 million bacterial genes. That makes it 10 times. So, since most body functions are regulated by genes, so the source of gene in our body, the cellular genes are only 20,000, but bacterial genes are 100 times more, 2 million. That of course means that there could be significant influence of these genes in our body function. So it is at least plausible that these bacteria have significant role in our body mechanism. Then if we just consider DNA, now this is actually a very, very interesting fact. If we collect total DNA of a human, 99% of that DNA is bacterial and only 1% is human. Look at this. So 99% genes in any normal human is actually the bacterial genes, not human. So if you weigh all these bacteria collectively, a total weight is almost equivalent to human brain. So no wonder our brain has got such a strong influence of our day-to-day activity. So it's no wonder that microbiota also has a significant role because it is like as big an organ as brain. So human microbiota is like an essential organ. I hope you found these figures very exciting. Now, microbiome as a second human genome. Now, let me actually clarify this word. Microbiome is collective genetic material of bacteria. If you take all bacteria and you take out their genes, a collection of those genes would be called microbiome. Whereas all the bacteria themselves taken collectively will be called microbiota. And human genome means all the genes which we inherit in our cells collectively that we call human genome. So this is the picture of genome, our human genome. And I think I'd like to draw your attention here. There is 99%, 99.5% homology. Like somebody sitting in Karachi is 99.5% similar to another person sitting in Shanghai or Chile or New Zealand. And there's only 0.5% diversity. All right, so most of the genome in all human is the same, 99.5. So we are different only by 0.5% as far as human genome is concerned. But when we consider microbiome, only 5% homology. So somebody sitting just next to you has got only 5% similar microbiome as you have. There is 95% diversity. So I think what makes you unique person is actually your microbiome because it is 95% typical of you, whereas 5% is generic, which is present in every person. So I think very interesting figures that what makes us unique is actually microbiome. More is microbiome as compared to genome. Every individual has a unique signature microbiome. Like we can identify anybody from his DNA, and it is definitely possible to identify a person from his microbiome. It may not be practically possible at the moment, but of course theoretically it would be possible to identify any person from his fecal matter. If you find his fecal material, you will be able to identify the person. So this is unique signature microbiota. This is because of unique microbiota we have in our intestine. If we count genes, a human has just over 20,000 genes. Our human genome has been completely outlined, and we know which gene does what, and we have only 20,000 plus genes. But and imagine a plant which has got very simple function. The rice plant, which is just like a plant, no big function like humans, it has got 40,000 genes. But the difference is that we have many bacterial genes, like 2 million microbial genes. So I think these 20,000 probably in harmony with the 2 million genes, they work together. That is how our very complex body functions are performed. So if you compare with the rice plant, it is not just a human gene, it is if you have to count these microbial genes also to have a fair comparison. So based on this new research, a consortium was made, the 7th International Human Microbe Consortium meeting, 26th, 28th June, Ireland, but it was actually constituted, I think, way back in 2007. They had annual meeting, they're doing research, and they are gathering information and seeing how this new science can translate into new tests, new treatments. So very interesting science. And the latest one was 8th Congress, and this was, I think, I can't see where was it, but this is also one of the meeting in this regard. Now, methodology, how do we study microbiome? This is interesting because conventionally we just used to see bacteria under microscope and with staining, H&E staining and culture. Now, I would like to mention here that this technique cannot pick up bacteria more than 20%. So 80% of our gut microbiome, they cannot be cultured. They cannot be stained with the conventional methodology. So they cannot be identified with this technique. There is a new science and there is 16 sRNA gene sequencing. This is newer generation testing. These are very, very characteristic RNAs of every bacterium. So scientists have developed a methodology where they can identify a 16S RNA of a particular E. coli, which would look very similar to another E. coli, but based on this 16S RNA, that bacterium would be characterized as a unique type of E. coli. So that is why we have found so many species. Another method of studying these bacteria is the simulator of the human intestinal microbial ecosystem. Scientists have developed a system which very much is similar to human gut. So bacteria grow there, more bacteria can grow, but I think most of the data is based on 16S RNA, rRNA gene sequencing. This is called SHIME, and of course, these days the apps and the handheld devices, they all play some role. So iChip and iTip, you can figure out what they mean. Some applications they have been developed which can be used to identify these organisms. So this is actually to show you that there is extreme diverse population which is based on this 16SR RNA gene sequencing. On microscopy, they would look very similar, but actually they are different. That is what makes us different. That is what makes person A different from person B. Now, let me examine two things. First, how host cells, like our cell, which we have already mentioned, only 10%, and microbiota, which is 90%, they interact. They, of course, human cells, they have interaction with the microbiota both ways. Humans, they affect microbiota and microbiota affects human, and both are affected by environment. So this is a complex interplay. That is what can sometimes changes and causes what we call dysbiosis, and that can lead to certain disease formation. So we will examine both how host and microbiota interact. How we, the host, affect our microbiome. This is the gut microbiota. So number one, mode of birth. This was very briefly mentioned. The method of the mode of birth determines our gut microbiota. Vaginal delivery versus cesarean section. Diet, especially the children in the early childhood, whether the baby was breastfed or was formula fed, and whether in the early childhood, whether somebody eats mostly plant-based diet or meat-based diet. And of course, plant-based diet is very favorable for healthy microbiota, and meat is relatively less healthy. But definitely it has been shown in the world that those populations where plant is a primary diet, their microbiota is different than those with Western societies which are mostly meat eater. Then antibiotic are one of the worst things human has invented for themselves. Actually, not antibiotics are great medicines, but since they are abused in, I think, 95% of the cases, antibiotic is used when it is not necessary, actually, especially in the childhood. So these antibiotics can affect our gut flora. So this is another way we the humans can affect our gut flora. Drugs which we take, like proton pump inhibitors, they reduce our acid. Metformin, which affect our gut motility. Statins, again, and then laxatives. These are just few of the many drugs which can have effect on our gut flora. Then diseases, inflammatory bowel disease and irritable bowel syndrome. Both people, the people with IBD or IBS, their gut flora is different than otherwise healthy people. Now, it would be hard to say whether they have developed this disease because of the change in the microbiota or their change in microbiota is secondary to their disease. We will examine this in my next lecture, but there is association between microbiota and inflammatory bowel disease and irritable bowel syndrome. Sleep, imagine just sleep, our sleep patterns and can affect our microbiota. Somebody travels from uh, Asia to Europe with 5 hours difference, time difference. There will be change in his circadian rhythm and there will be also change in his gut flora. And exposure to pets and others, generally especially in our culture, it is considered that children should not be exposed to pets. At least they should not interact with them closely, but we know that exposure to pets and other animals, especially in the childhood, is very favorable and they can lead to promotion of healthy gut flora. So these are some of the factors in humans which can affect our gut microbiota. Now let's see the other side of the picture, how our microbiome affects us. This is gut microbiota, and let's examine how that gut microbiota can affect our body functions. We are still talking about gut microbiota and health. In the next lecture, we are going to discuss various diseases. Like bacteria, they play a very important role in digestion of plant-based foods. You know, plant-based food, they have cellulose, and our stomach and pancreatic and intestinal enzyme, they cannot digest cellulose. It is the bacteria present in the large gut which digests some of these plants. Now, in animals, this is actually up to 30%. In humans, it may be about 10%. So, bacteria definitely play an important role in the digestion of our food, especially the plant-based food. Then there is vitamin synthesis, folates, folic acid, vitamin K, biotin, riboflavin, and cobalamin. All these vitamins have been known to be produced by our gut bacteria. These are all essential things, but what an important role our bacteria are playing for us. Then defends against colonization by opportunistic pathogen. And this is perhaps the very important function. Our gut bacteria, they are like our policemen, and any invading bacteria, any pathogen is first treated by our bacteria. Mostly our bacteria, they do not allow these opportunistic pathogens to grow and to cause disease. Then immune modulation, I have already discussed very briefly in the second or third slide that exposure to foreign world, exposure to dirt, exposure to filthy things actually enhance the development of our immunity. And it has been observed that in those environments where there's a super hygienic environment, actually some diseases like allergic disorders, like asthma, and these conditions can be actually atopies, they are more common. So gut microbiota, they help our immunity modulation. Then energy production, very interesting, 5 to 10% of the daily need, our calories, 5 to 10% of the energy is harvested from that food which our enzymes cannot digest but bacteria digest is for us. So up to 5-10% of calories, they come from bacterial source. Then production of these small chain fatty acids. There are many of them. Uh, they have very important role in our body metabolism. They actually are small fatty acids. They are absorbed from the gut. Even the healthy gut will allow to these fatty acids to be absorbed, and they play important role in different body functions. Then bacterial translocation. Now this I do not mean the true bacteremia, which we normally know is very, very bad. Somebody develops bacteremia is usually very sick, but this means some bacterial components, not necessarily whole bacteria or maybe the whole bacteria, but they're not pathogens. They circulate through the blood and then they go to liver, they go to spleen, they go to various organs. Although this science is still developing, but we know that these things, they go to different organs and they produce various types of metabolic effects. Some of them are very, very useful in maintaining our normal health. So the most important determinant of our gut flora is our diet, and generally it is considered that vegetable and plant origin diets are healthier diets. So I think we should consume a lot of vegetables and fruits to promote our healthy microbiota. And unfortunately these days the younger generation, the children, they do not eat fruit, they do not take vegetables, they do not take salads. So I think we need to promote this culture to avoid these diseases. We need to consume more vegetable based foods because that is known to promote healthier microbiome. Now, further studies, these are three important papers I have referred to. This one is free, the Human Microbiota in Health and Disease, and you can access this one. This one you have to buy, Human Intestinal Microbiome in Health and Disease. This one is other current understanding of the human microbiome. These are all available, and if anybody is interested to learn more about this new and interesting science, he is referred to these papers. There are of course many other. When I typed the word human health and microbiota, I pulled I think something like 16,000 papers. That indicates the research interest in this topic. I would dedicate this lecture to my mentor, my teacher, my friend, late Professor Faisal Madud. It was because of him that I developed interest in this topic, and I will be actually looking forward to see you in my next lecture, which is on microbiota and human disease. So this is Professor Aziz-ur-Rehman from Medurstand. Thank you very much for your for watching this video. Thank you.